Hello, my name is Thibaut Meyer, and I'm a faculty here in the Department of Biochemistry and Molecular Biology. Our lab is actually located in the Michael Smith Laboratories, that is located centrally in the campus and is just behind me. And is also there a great opportunity for reminding everybody that UBC is located on ancestral, traditional and unceded territories of the Musqueam peoples. And here we have this very nice totem from Brent Sparrow Jr. located just close to us. So I'm going to bring you inside our lab now and show you a little bit about the work that we do here. So we're going to enter our main lab space where you can see some people, or everybody's wearing their mask, keeping their social distances. We mostly have small equipment. This is where most of the work is done. In the back, you see some additional equipment such as incubators, freezer, etc. I'm going to bring you now to our cell culture room that was recently renovated, when you can see our incubator and our biosafety cabinet. And finally, this is our mass spec room, in which we have several equipment that we share with Leonard Foster. So now I would like to spend the next 4 to 5 minutes to talk a little bit about one of the main projects for which we have, for which we like to recruit new students. So we're really interested in protein homeostasis, and more specifically how misfolded proteins can be recognized in the cells and targeted for proteasome degradation. So typically in the cells, most proteins can reach their thermostable native state, but once in a while, because of some error during transcriptions, translations, or maybe due to some mutations, the protein cannot fold to its native states. And what that happened when the chaperone machinery cannot help that proteins to fold to its native states, the cell has to recognize that and target that proteins for proteasome degradation. So what we know is that the ubiquitin proteasome system is implicated in the turnover of many of these cytosolic misfolded proteins, but we don't know yet exactly which E3 ligase is implicated specifically in mammalian cells. So we've done a lot of work in years in the past 10 years where we've been quite successful, but in the past 3 years we spent a lot of energy to establish all the system in mammalian cells where we're a little bit more interested to look at how the system works. And so our starting point was to try to identify the panels of mutant proteins, of mutant cytosolic proteins that are becoming degraded specifically when they have a single point mutations. So we mine a database of over 2,000 disease specific mutations and look specifically at cytosolic protein that we thought potentially might be degraded by the proteasome, and now we have a panels of about 20 model substrates that behave that way. And so the way we do the work now is actually we use the GFP moieties on these proteins so that we can monitor their fluorescence within the cell. So we don't need to do Western blot to see the expression level. And so we can stably express these wild type or the mutant variants in cells. We typically use HEP293 cells. And when we do so, if we look at the wild type proteins, we can see a pretty high fluorescence in the cells. In contrast, when we look at the mutant proteins now, we see a much lower fluorescence. It's because this protein is not very stable in the cells. We can add an inhibitor that inhibits the proteasome or that inhibits the E1 enzyme that is required for the ubiquination to occur. And when we do so for a couple of hours or several hours, what we see is that now we have the accumulation of the mutant proteins. But the wild type protein typically remain unchanged. That is a clear demonstration that the ubiquitin proteasome system is used to target these misfolded protein for degradation. But we don't know yet which E3 ligase is doing that work. So we assess a couple of candidate genes. We knock them out by using CRISPR-Cas9 approach, but when we did so, it did not influence the stability of the mutant proteins. So this was very disappointing for the students who did the work, but it's very exciting for us for the lab because it's telling us that we still don't know what is the E3 ligase that recognize these misfolded proteins. And so now we really want to screen using different approach to identify the candidates. And so one of the approaches with our collaborator, Dr. Mikko Tapale at the Donnelly Center at the University of Toronto, and he has a CRISPR-Cas9 library specifically of many of these E3 ligases. And so using his library, they are screening or reported to see which of these knockdown can stabilize the mutant proteins. And in our side, we start to do more proteomics to identify which proteins interact with the misfolded protein but not with the folded protein. So using this bio-ID approach, what we hope is to identify both chaperone and E3 ligase that are important for the turnover of, or preventing the turnover of these misfolded proteins. And so once we have identified the E3 ligase, the next step will be then to better understand how that E3 ligase can recognize the misfolded protein. Can it directly interact with hydrophobic patches that are potentially more exposed with the substrate? Does it recruit a specific chaperone proteins that can recognize these misfolded proteins? That would be sort of like the next frontiers that we would like to explore, hopefully maybe with you. And so if you are interested, join us and join me in one of the breakout rooms so that we can talk more about how we want to move forward with that project. So thanks for listening.